All right, how about transduction then? To understand transduction, we have to talk a little bit about viruses. Remember, viruses are very simple organisms, just a chromosome, viral chromosome surrounded by a protein shell called the capsid. And they can't reproduce themselves. They have to get inside other cells and trick those other cells into reproducing them. And viruses can be very cell-specific in terms of some viruses only affect human beings, and some might only affect liver cells in human beings. Others might only affect certain species of plants, that sort of thing. Well, there are certain viruses that only infect bacteria, and those viruses are called bacteriophage, viruses that will only infect the bacteria. And so that's what we're going to see here. Here's this bacteria sitting there minding its own business, and here's a bacteriophage that comes along, and it gets inside the bacteria. And then, like all viruses, it will now try to trick the cell into reproducing itself. The viral chromosome exits the capsid, and then the bacteria says, oh, there are some genes. The bacteria doesn't know that these are viral genes, and so the bacteria says, I'll start expressing those genes. But of course, those are viral genes, and so therefore, things like making new capsid proteins and genes for making copies of the viral chromosome. And then those new copies of the viral chromosome load up inside the capsids like that, and that's how the bacteria has reproduced the virus. And usually this happens to such an extent that the host cell gets destroyed. The bacteria production fills it up so much that it essentially bursts. Now, as the bacteria is manufacturing the viruses and the bacterial cell is starting to disintegrate, what can sometimes happen is this: a piece of the bacteria's own chromosome can break off, and just by random luck, that can get packaged inside a viral capsid, like this. That's not the point of the capsid, but it can happen sometimes. And so look at this weird thing that gets produced here. You have a viral capsid, a protein shell, surrounding and protecting that piece of DNA, but it's not the viral chromosome, it's just some genes from the bacteria. Well, when the bacteria bursts, these viruses, so to speak, exit and go drifting outward, and then they're going to infect other bacterial cells. But this one right here is kind of special. Let's follow that guy. So here he is drifting here, and so when that one infects this other bacterial cell over there, notice that it's not going to cause the lytic cycle or anything like that, because to produce more viruses, we would have to have viral genes on there, and those aren't viral genes, they're genes from the other bacteria. And so when that little bit of DNA pops out of the capsid, it can just become more genes inside the new bacteria. But notice what's happened here. The new bacteria has picked up genes from that other bacteria by way of a bacteriophage capsid, and that's what transduction is. When bacteriophage virus is what transfers the genes between bacterial cells. The abnormal bacteriophage infects another cell. That bacteria will receive genes from the old virus. Alrighty. All right. Well, the third method of gene transfer is called conjugation. And as we go through this one at first seems a lot like sexual reproduction, but again, it's not really sexual reproduction. It just sort of has that air about it. And let's start with this part right here. Some bacteria, not all bacteria, but some bacteria have a group of genes called the F factor. Here I showed the F factor on a plasmid, which they oftentimes are, but they don't have to be. The F factor cluster of genes can also be on the main chromosome. Any bacteria that has this F factor cluster of genes is called an F plus bacteria, meaning they have the F factor, or they sometimes call them HFR, which stands for high frequency of recombination bacteria. And any bacteria that do not have the F factor are called F minus bacteria. All right, and as I said here, the F factor cluster of genes can be on the bacteria's chromosome or one of its plasmid. It could be either spot. That's right, yeah, it could be there. Okay, now, so all genes encode proteins, and proteins have some sort of function. What do the F factors do? Well, the proteins that are encoded by this F factor encode a group of genes that can form something called a sex pilus. And this is essentially a grappling hook that the bacteria, the F plus bacteria, can use to grab hold of another bacteria, pull it close, and then transfer the F factor genes to that other bacteria. So I'm saying when these genes are expressed, it causes proteins to be made in the bacteria. Think of these as a protein structure. It causes some proteins to be made in the bacteria that form something called the sex pilus. And that's the grappling hook. It grows out of the F plus bacteria and can essentially reach out, snag hold of another bacteria, an F minus bacteria, and pull it inward. And this sex pilus thing is not only a grappling hook, it's actually tubular. It's a hollow tube, and so it can form what's called a cytoplasmic bridge between the two bacteria. So their cytoplasms that are linked together through the sex pilus thing. The F factor genes not only encode the sex pilus, they also encode enzymes which will copy the F factor and transfer it through the sex pilus into the F minus bacteria. There's a copy, the F factor genes have copied themselves. They move through the sex pilus. They go into the new bacteria. And so that new bacteria is no longer an F minus bacteria. It has now become an F plus bacteria. And so this new bacteria, now that it's F plus, will be able to do the same thing. It will be able to manufacture its own sex pilus, which will allow these F factor genes to then transfer themselves to another bacteria. And again, this is really weird. We might wonder, well, are these F factor genes doing any of the bacteria any good? And at first it seems like they're not, because it doesn't really benefit this bacteria to have those genes. I mean, this bacteria, if anything, has to expend resources to manufacture the sex pilus and to make enzymes to copy this F factor thing. It has to expend resources. So I don't think it's actually doing this one any good. And so it's almost as if the F factor is sort of a selfish group of genes that are just using the bacteria as a host to copy themselves. Because notice this thing is reproducing itself. And there was only one copy here of the F factor genes, and now there are two copies of it. So virus is not the right word, but it is almost like a selfish bit of DNA that's just trying to copy itself from bacteria to bacteria. Okay, so that's what the F factor is all about. Now, let's go back in time for a second. Here we go. Here's that first bacteria. When the F factor genes are on the bacteria chromosome, occasionally what will happen is when the F factor is copying itself, well, there's the sex pilus it's making. But sometimes what will happen is when the F factor is copying itself, it will also copy some of the surrounding genes, some of the nearby genes on the bacteria chromosome. Like that. Here you can see some of the bacteria genes came along for the ride accidentally. And so when that F factor moves across the sex pilus to the F minus bacteria and ligates itself into that bacteria's chromosome, that bacteria is picking up some new genes that could potentially be of benefit to it, since those might be beneficial genes from that the original bacteria had. So let's reevaluate the question. Does having the F factor do a bacteria any good? At least for the recipient bacteria, you might argue it might, because sometimes that will mean that it might be getting some new genes from another bacteria, and those potentially could be improvements of beneficial genes to the recipient bacteria. All right. You guys are quiet today. I guess that's all right. Okay. Well, if there are no questions on conjugation and transformation and transduction, we're going to switch gears now. Up to this point in today's lecture, I've been emphasizing how genes can move from one bacteria to another. And now we're going to switch to a different topic, the expression of genes in bacteria. And remember, that means how the bacteria make mRNAs from the genes, and those mRNAs become proteins that the bacteria is going to use. All right, and bacteria, actually all organisms, have to be able to regulate gene expression, meaning that sometimes it's appropriate to be expressing a gene. In other words, you need the protein, but other times you may not need that protein. I guess a good example in human beings is this. The cells that form the lining of your stomach make digestive enzymes to help you break down your food. And when you've eaten some food, you need to express those digestive enzyme genes to break down your food, but in between meals, it would be wasteful of you to be generating a bunch of enzymes that you don't need at that moment, and so you want to turn off expression of those genes. Well, the bacteria the same way. Sometimes they want to express genes, sometimes they don't. And to give an example of that, I'm going to talk about the bacteria that live inside your intestines. Here's your large intestine here, and there's a whole bunch of bacteria. Matter of fact, there's a bunch of archaea that live in there also. And they get some of the nutrients that we can't digest. They essentially feed on bits of nutrients that are left inside the feces. And so I'm going to use them as an example of bacteria whose environment keeps changing, because what exactly nutrients are available to them depend on what you ate for your last meal. If you eat a carbohydrate-rich meal, there's going to be lots of carbohydrates in their environment. If you eat a protein-rich meal, there's going to be lots of amino acids there. If you eat a fatty meal, there's going to be lots of fatty acids and glycerol in there. But these basic principles that bacteria have to change their gene expression because their environment changes is really what I'm illustrating here, not something about just the digestive system. The digestive system is just handy to illustrate a changing environment. Alrighty, so let's say that you eat a protein-rich meal. What would that be? A bunch of steak or soybeans or something like that. Proteins are made out of amino acids, and as you digest the proteins, you break them down into their individual amino acids. And one of the amino acids is called tryptophan, whose three-letter abbreviation is TRP. Oh, what happened there? I think I brushed the projector off button. Great, that's going to take 5 minutes to warm itself up. Well, that's fine. Anyhow, I'll use this bacteria until it comes back on. So when you've eaten a meal that contains a lot of proteins, these bacteria are going to be surrounded by a lot of amino acids. in their environment, such as the amino acid tryptophan. All cells, including bacteria, need the amino acids to build their own proteins out of. And so this bacteria is going to want to import that tryptophan to help manufacture its own proteins. So it actually has transport proteins, so there we go, to bring the tryptophan inside of itself. And that's handy for the bacteria. It doesn't have to manufacture this tryptophan amino acid for itself because you are providing it for it in the environment. Has anybody heard of tryptophan amino acid? I remember there's a, I don't know if urban myth is the right word, but whenever you eat a lot of turkey for Thanksgiving, people always get very sleepy. And somebody claimed at some point that it's the tryptophan in the turkey meat that makes you get sleepy. I don't know if there's any truth to that, but just curious if anybody else had heard that. So maybe we get sleepy bacteria or something, I don't know. Anyhow, so the bacteria import the tryptophan. Now, let's say there's the tryptophan's going inside. Let's say then that you change your diet and the next time you eat, you don't eat any proteins at all. Maybe you eat, I don't know, potatoes or something like that